Today we are going to learn about citric acid cycle. Citric acid cycle is also known as Krebs cycle or tricarboxylic acid cycle. It is the most important metabolic pathway for the energy supply to our body because around 65 to 70% of the ATP are synthesized in the Krebs cycle. Citric acid cycle involves the oxidation of acetyl coenzyme A to carbon dioxide and water. So, citric acid cycle is the important pathway because around 65 to 70% ATPs are synthesized in the citric acid cycle. The name citric acid cycle is used because the three acids, that is tricarboxylic acids like citrate, cis-aconitate, and isocitrate, are participate in this cycle, and therefore it is called as the tricarboxylic acid cycle or TCA cycle. The TCA cycle is the central metabolic pathway of the carbohydrate metabolism because it connecting to almost all the pathways in our body directly or indirectly. That's why it is called as the central metabolic pathway. The citric acid cycle was proposed by the scientist Adolf Krebs in 1937 based on the studies of the oxygen consumption in the pigeon breast muscle. The cycle is named in his honor. Nobel Prize he got for physiology and medicine in 1953. The enzymes of the TCA cycle are located in the mitochondrial matrix in close proximity to the electron transport chain. Because of this, the synthesis of ATP by the oxidative phosphorylation occurs without any hindrance. The TCA cycle, if we see, it involves the starting material for this cycle is the acetyl coenzyme A, which contains the 2 carbon atom, and which combines with the oxaloacetate, which contains the 4 carbon atom, and by the combination of acetyl coenzyme A and the oxaloacetate, we get the production of 6 carbon tricarboxylic acid citrate. So this is the first step of the TCA cycle. The reactions of the TCA cycle we will see one by one. These are all the reactions of the TCA cycle. If we can able to see here, pyruvate is the end product of the glycolysis, and the starting product of the TCA cycle is acetyl coenzyme A. So this we can say this is the intermediate step between the glycolysis and the TCA cycle. The pyruvate is converted to the acetyl coenzyme A by the use of enzyme pyruvate dehydrogenase, and here the NADH molecule is produced. And also the coenzyme A is added at this step, and after that the acetyl coenzyme A is produced from this step. Now this 2 carbon atom acetyl coenzyme A will react with the 4 carbon atom oxaloacetate by the use of enzyme citrate synthase, and coenzyme A is removed from this step, and we get the first acid formed at this step, that is the citrate. This is the first step of the TCA cycle. Then after citrate, we get the formation of cis-aconitate and isocitrate by the use of enzyme aconitase. After isocitrate, then this is the important step. Isocitrate is converted to the oxaloacetate, and at this step, the first NADH molecule is produced of the TCA cycle by the use of enzyme the isocitrate dehydrogenase. This enzyme is also important, and after isocitrate, this enzyme is only involved after oxalosuccinate, we get the formation of alpha ketoglutarate. Enzyme is same. So if we can say that by the use of enzyme isocitrate dehydrogenase, the first NADH and first CO2 molecule is produced in the TCA cycle. From isocitrate, if we directly say alpha ketoglutarate is produced, and then at this step, the first NADH and first CO2 molecule is produced in the TCA cycle by the use of enzyme isocitrate dehydrogenase. Then after alpha ketoglutarate, the enzyme used is alpha ketoglutarate dehydrogenase, and we get the formation of succinyl coenzyme A. At this step, the second CO2 and second NADH molecule is produced in the TCA cycle. The step is alpha ketoglutarate to succinyl coenzyme A. Enzyme used is the alpha ketoglutarate dehydrogenase, and the second CO2 and second NADH molecule of the TCA cycle is produced at this step. After succinyl coenzyme A, then we get the formation of succinate by the use of enzyme succinate thiokinase, and at this step also the 1 GTP molecule is produced. First GTP molecule is produced in the TCA cycle. After succinate, then we get the formation of fumarate. At this step also we get the first FADH2 molecule is formed in the TCA cycle by the use of enzyme the succinate dehydrogenase. After fumarate, we get the formation of L-malate. fumarase enzyme is involved here, and after malate, again the oxaloacetate then regenerated in the TCA cycle after malate by the use of enzyme malate dehydrogenase. At this step, the third NADH molecule is produced and the oxaloacetate is regenerated, and then this oxaloacetate again enters into the TCA cycle. It again combines with the acetyl coenzyme A, and again it will form the citrate, and the further cycle will then continue. So this is all about the TCA cycle. If we can see the steps of the TCA cycle, in the first step is the formation of citrate, as we have seen now. Okay. Then after in the second step, then the citrate is isomerized to the isocitrate by the use of enzyme aconitase. Okay. Then in the fourth and fifth step, as we all know, the alpha-ketoglutarate is formed, and this is an important step because here the formation of NADH and liberation of CO2 occurs at this step. Okay, and the enzyme used here is the isocitrate dehydrogenase. This is the fourth and fifth step. At the sixth step, then the formation of alpha-ketoglutarate to the 16 coenzyme A occurs, and at this step the second NADH is produced and the second CO2 is liberated. Then in the seventh step, the formation of succinate. At this step, the GTP molecule is produced. Then after succinate to fumarate, there is also the formation of the production of FADH2 molecule occurs. Then by the use of enzyme succinate dehydrogenase, then in the 10th step, we get the formation of the regeneration of the oxaloacetate, which again then enters into the citric acid cycle. So these are all the 10 steps involved in the TCA cycle. Now, how many ATP molecules are produced? This is important. The energetics of the TCA cycle. We all know that the 3 NADH are produced in the TCA cycle. One FADH2 is produced, one GTP molecule is produced. So if we calculate the overall production of the ATP, we can see here glycolysis we have already covered. This is the intermediate step, pyruvate dehydrogenase. From these 2 NADH molecules are producing, so 6 ATP are formed at the intermediate step. Then in the actual citric acid cycle, the first enzyme isocitrate dehydrogenase, from this the 2 NADH molecules are produced, because I have told you that 2 pyruvate molecules are produced in the TCA glycolysis. So from 2 pyruvate molecules, 2 NADH are produced here. 6 ATP are formed. Alpha-glutide dehydrogenase enzyme, 6 ATPs are formed. Succinate thiokinase, 2 ATPs are formed. Succinate dehydrogenase, 2 FADH2 means 4 ATPs are formed, as 1 FADH2 will give the 2 ATP molecules. Then malate dehydrogenase, we get 2 NADH again, then 6 ATP. So total, if we can calculate, this will be 24. Okay, so this is the 8, intermediate 6 and 24. Total, if you can calculate, so around 38 ATP molecules are produced from one glucose molecule. And in the anaerobic condition, the 2 ATP molecules are produced. But if we can see, this is for the 2 acetyl coenzyme A. If we want to calculate the 1 acetyl coenzyme A, then we get only 12 molecules. So we can say that from 1 acetyl coenzyme A, we get 12 molecules of ATP. So in the TCA cycle, total 24 ATP molecules are produced. Okay, as what I am telling you, see, these are the 2 pyruvate molecules are formed from the glycolysis. So 2 acetyl coenzyme A will produce. So whatever NADH will produce at each step, that will be, there will be 2. Here 2 NADH will produce, here 2 NADH will produce. So that's why I have the calculation is like that here. Okay, so like that, for isocitrate dehydrogenase 2 NADH. 2 FADH2, 2 NADH, because 2 acetyl coenzyme are there. So total 38 ATP molecules are produced in the TCA cycle. These are the inhibitors of the Krebs cycle. Now, the important point here, the Krebs cycle is amphibolic in nature. Because why it is amphibolic? Because here the both catabolic and anabolic pathway occurs in the Krebs cycle, and that's why it is called as the amphibolic in nature. The Krebs cycle is the amphibolic in nature. So this is all about the Krebs cycle.